So far we have talked about organismal aging and organismal aging phenotype. Let's go a little bit deeper and look at the cellular level. Let's start with cellular senescence. In the previous section, we've talked about some organismal features of aging organisms. But all these macroscopic features, if you will, must derive from some cellular and molecular origins. We've already met some molecular aging pathways, especially the IIS and the ROS production pathways. But in this module, we will focus entirely on these cellular and molecular pathways. One of the earliest insights into cellular aging mechanisms came when Leonard Hayflick discovered that cells grown in culture are not immortal, and instead only divide for a set amount of cell division, usually around 50 cell divisions. This is now known as the Hayflick limit. The Hayflick limit is often attributed to the shrinking of the telomeres. These are protective caps at the end of eukaryotic chromosomes. With each cell division, telomeres lose material and shrink until chromosomes become so short that they are no longer capable of performing their proper function, and further division becomes impossible. Interestingly, an enzyme called telomerase can restore damaged telomeres back to their elongated state, thus resetting the cellular telomere clock. For a long time, telomeres and telomerase were thought to be key in anti-aging interventions. But as so often, those hopes were dashed when telomerase transgenic mice did not live longer than control animals. The fountain of youth is still elusive. In any case, this inability for further divisions can also occur independent of telomere status and represents a true biological phenomenon, that is, cellular senescence. It is important to differentiate cellular senescence from our common use of the word senescence, which simply means old age. Cellular senescence is a defined set of biological parameters that describe the functional state of a cell. As such, it has to be differentiated also from quiescence, a similar state of cellular inactivity, but characterized by a different set of markers. To understand this, we have to look at how a cell lives. Cells go through distinct phases in their life. After a successful mitosis, the cell enters interphase, the time between cell divisions. Interphase can be further subdivided into a growth phase, G1, during which the cell grows and creates energy reserves. This is followed by the S or synthesis phase, during which DNA is duplicated in preparation for another round of mitosis. S phase is followed by another brief G phase, during which the final preparations for mitosis are made, and finally mitosis is executed. Interestingly, cells may exit the cell cycle and not perform growth and further divisions. This is particularly important for multicellular organisms that require a fixed number of cells to form organs. Thus, cells enter so-called G0 phase and stop cycling. These cells in G0 are so-called quiescent cells, cells that do not further divide until activated by mitogenic signals. Cancers often develop because quiescent cells are erroneously activated and start dividing in an uncontrolled fashion. The most important distinction between quiescent and senescent cells is that senescent cells are unable to re-enter the cell cycle. While many of the molecules that induce a cell to enter quiescent state are the same as the ones for a senescent state, a senescent cell becomes altered in function and produces many molecules that quiescent cells do not. This senescence-associated secretory phenotype, or SASP, is accompanied by a morphological change to a large and flattened out cell. SASP often involves the secretion of pro-inflammatory cytokines into the local cellular environment, which may in turn contribute to accelerated cellular aging of neighboring cells. Senescent cells are remarkably resilient, often persisting for years in a tissue context. In vivo, it is frequently difficult to detect senescent cells, making a clear statement of the contribution of cellular senescence to organismal aging problematic. Some markers that are used to identify senescent cells in vivo are the senescence-associated beta-galactosidase, or SA-beta-gal, ARF, or P16-INK4A. None of these markers in isolation have proven to be completely reliable, though. Current research is very much focused on developing better markers, or panels of markers rather, to truly get a better appreciation of cellular senescence in the organismal context and in organismal aging. Intuitively, it makes sense. Senescent cells do no longer contribute to renewal potential of an organ and have potentially detrimental secretory phenotypes. However, the Ross theory of aging also made intuitive sense, yet has not been convincingly shown to be all that important to aging phenotypes. How senescence is induced in a cell is still unclear and the subject of intense research. One possible explanation revolves around the protein p53. p53 is one of the most famous proteins out there, and in fact, the most studied molecule ever, with over 50,000 publications to his name. p53 is mutated in over 50% of all cancers, highlighting its important role as a tumor suppressor protein that, when active, inhibits the formation of cancerous cell growth. Because of that function, it has been nicknamed guardian of the genome or the king of tumor suppressors. p53 is involved in cellular senescence because it is activated when DNA is damaged. Under normal, healthy cellular conditions, p53 is constantly made, but it gets immediately degraded, leading to no net p53 activity. Once DNA damage is encountered, and by that term, I will also include shortened and degraded telomeres, p53 gets stabilized, meaning that its automatic degradation stops, and p53 can begin executing its activity. As one part of its function, p53 may activate the p21 protein, which leads to DNA damage repair. If the p21 pathway cannot repair this DNA damage, then the other functionality of p53 sets in. That is, to remove this damaged cell from the organism entirely. The easiest way to do so is to induce apoptosis, or cellular suicide, which kills the damaged cell, thus removing it from the organism, and therefore eliminating any potential for this damaged cell to develop into a cancer cell. The other way to deal with such a cell would be to induce permanent growth arrest in the cell as well as a functional reprogramming, thus, as before, eliminating the threat the cell may pose. Thus, instead of inducing apoptosis, p53 may induce cellular senescence. Another senescence-inducing pathway involves the genetic locus INK4A. This genomic locus is quite unique in that it encodes three different proteins called p15 or INK4B, p16 or INK4A, and ARF. The two INK proteins are inhibitors of cyclin-dependent kinases, which are proteins that control cellular progression through the cell cycle. Thus, if the cell cycle is stopped, we essentially end up with a senescent cell. These steps bypass p53 and can occur independently of p53 activation. However, it is important to note that the p53 and the INK pathways are not as clearly separate as this simplification implies. The third protein produced by the INK locus, ARF, in turn acts to stabilize p53 and leads to p53 activation, with all that entails. What is important to note here is that p53-dependent senescence is closely associated with DNA damage, either through mutagens, ionic radiation, oxidative damage, or telomere damage, while INK4A-dependent senescence is largely DNA damage independent and more closely related to mitogen activation, such as oncogene or growth factor signaling. Not only do different stimuli induce different senescence pathways, different cell types may also prefer one type of senescence over another. Interestingly, p53 and INK4A-dependent senescence pathways may furthermore lead to subtly different senescent phenotypes, including the precise nature of the senescence-associated secretory phenotype, the formation of senescence-associated heterochromatin foci, or SAHF. These are regions of permanently silenced genes, which often includes genes responsible for proliferation and growth promotion. And even the ability of a cell to possibly re-emerge from senescence back into the cell cycle. INK4A induction does not appear to allow that, while p53 induction may. In this case, p53 needs to be continuously active to maintain senescence, leaving open the possibility that if DNA damage has been sufficiently repaired, the cell may resume its regular function. It should be noted that the majority of the evidence of the senescence-inducing molecular pathway comes from in vitro studies. As mentioned earlier, it is very difficult to detect senescent cells in vivo, partly because of their relative paucity. Senescent cells may be cleared from the organism by the immune system, as has been suggested for senescent cells from the liver, or there simply may not be many in any given tissue section in the first place. It may also be an issue of being able to detect them. We did talk about the lack of senescent biomarkers, mostly that SA beta-gal, INK4A, and p53 are used to detect senescent cells. But those are not entirely exclusive to senescent cells and may appear in other cells as well, and only a combination of at least two of those is required to make a determination. The best example of senescent cells in vivo is in skin, where skin lesions called nevi are often associated with large amount of senescent cells and a strong SASP phenotype. These senescent cells may persist for decades in the skin, suggesting that at least for the skin, immune clearance of senescent cells is not a big consideration. Senescence in this example may thus be a mechanism to prevent UV-damaged skin cells to progress into a full-blown cancerous state and protect the skin and the organism from melanomas. As we have seen earlier, senescence can be induced by a variety of stimuli in different molecular pathways. In vivo, the biggest effect senescence may have is on proliferating cells, such as stem cells that are important for the body's self-renewal functions. If such a stem cell becomes senescent, it will thus fail to replenish the tissue with young new cells, leading to tissue cell loss and tissue dysfunction. Even the cytokines secreted by the SASP phenotype might contribute to tissue malfunction over time. With advanced chronological age, the cells expressing senescence marker molecules increase exponentially. This has been shown for a variety of markers, such as INK4A or telomere damage, in a variety of tissues, such as white blood cells or skin cells, and even in different organisms. What's more, this increase in senescent cell number is moreover associated with a loss of regenerative potential of organs, presumably by turning some stem cells into senescent cells. We know that stem cell numbers decline with age, leading to the inability to replace damaged cells or cells that were lost, which in turn negatively affects tissue function. Therefore, senescence may be contributing to tissue, organ, and organismal aging phenotypes by inactivating stem cells and tissue self-renewal functionality. In addition, the SASP further contributes to tissue dysfunction, in part by overproduction of interleukin-6, a cytokine with a major role in promoting cellular proliferation, presumably to make up for the functional cell loss caused by senescence. Just because senescence functionally disables cells does not mean it is all bad. As we have seen earlier, senescence protects the organism from the development of cancerous cells, such as melanoma cells. It furthermore disables immune cells, which prevents the development of autoimmune diseases, such as lupus. Senescence is thus an important organismal self-protection system, just like programmed cell death or apoptosis is, that functions to efficiently remove or at least disable aberrant cells. With such a clear correlation between senescence and tissue dysfunction on one hand, and tissue dysfunction and aging on the other hand, it is easy enough to speculate that cellular senescence may play a role in aging. As mentioned in previous studies, genome-wide association studies have identified the INK4A locus as associated with longevity, and many age-related diseases can be mapped to certain polymorphisms in this gene locus. But unfortunately, all this evidence is merely correlative. In addition, organisms that are considered postmitotic, such as C. elegans or fruit flies, that, in other words, do not self-renew their tissues, still undergo aging. Thus, cellular senescence may be a private aging mechanism as opposed to a universal or public mechanism that mainly affect organisms that rely on a large pool of self-renewable cells. Check your understanding. Define cellular senescence. Describe senescence phenotypes. Describe molecular pathways leading to senescence. Explain the normal function of cellular senescence and how this may directly contribute to aging.